Maybe Sandy, you can just talk a little bit about the basis for this, like why the improved genome was so important, and also kind of what it does, and kind of the reason why you wanted to put this paper together initially.
Okay, well, so the improved genome is only part of the story, right?
Right.
There's the gene expression story in
Yep,
three regions of
exactly.
the brain.
Yep.
But tying it to this new genome was unprecedented in terms of the quality of mapping RNA-seq reads. So
Yep.
being able to quantify the transcriptome,
Yep.
of course, depends on a genome. And the better the annotation and the better the genome, the better your understanding of differential expression in
Yep.
hibernation is going to be. And I guess I want to start by saying, I think I'm still totally devoted to the concept that phenotype comes from expression of genes. And therefore, if we want to understand the hibernating phenotype, we need to understand the differential pattern of expression that goes with the phenotypic changes that are characteristic of hibernators. And you know what's important for human medicine out of that, I can think of the two most important things are resilience of tissue to things like cold and ischemia reperfusion damage, and then also just the whole idea that you can reversibly metabolically suppress the function of a cell. I mean, you just take metabolism down to nothing and that's fine for a while and then restore it. And so these things are, I think, incredibly important, and the lessons we can learn from them are incredibly important. And um, so understanding differential expression is required for that. And without a genome, I mean, I think the two reasons the genome is really important, and the better the annotation and the better our understanding of where the genes are and what the genes are and how they're expressed,
Right.
the closer we're going to get to figuring all this out. And so before, the genome was broken into a gazillion little bits, and
Right.
genes were broken by that too. And so you actually didn't measure the expression of a single gene because it was broken across two contigs.
Right.
So getting rid of that helps a lot in terms of understanding the gene structure. So in past gene expression studies, the thing that's really different about this one, aside from tying it to a better chromosome level genome, is that we have these time points that are much more rigorously collected. So other people have studied differential expression with the same goals, but they randomly took samples across a torpor bout and randomly took samples across the warm period, and that all averages out and they won't see anything interesting.
Yeah.
So even our study is undersampled, no question about it, but at least the better sampling gives us the chance for the first time to really segregate gene expression changes that are linked to the seasonal phenotypic
Yep.
reprogramming
Yep.
from the ones that happen as part of the torpor arousal cycle. And even though I think that was foreshadowed in the paper Katie did earlier on brown adipose tissue,
Yeah.
it was never hitting you over the head in the same way it does in this paper. I mean, the heat maps that we present make it crystal clear, you know, which genes are seasonal expressed and which ones are cycling as a result of the torpor arousal cycle.
What was the sort of the most exciting thing, or what was maybe the most surprising thing that came out of the study that maybe you weren't expecting?
I mean, I think the large number of differentially expressed genes
Yeah.
was a huge surprise.
Mm-hmm.
Although maybe it shouldn't be, because temperature has a profound effect on transcription. And then we know, again, this was it was demonstrated actually in Katie's brown adipose tissue paper
Yeah.
about the stability of RNA control in the
Yep.
torpor arousal cycle is almost certainly really, really important. And so one of the things we found was this large cohort of RNA binding proteins that
Yep.
are cycling in the torpor arousal cycle. And some of them actually lead to rapid degradation and others lead to protection. And so there's this complex interplay between transcription and stability that are characteristic of the, I think, just extremely rapid gene expression dynamics that must characterize that little short period of euthermia.
Such a short time, right?
Yeah, and I'm pretty sure that, you know, there are components of cell cycling being restored, you know, so like very early, the first thing that has to happen is if there's going to be cell division, you need to get that started. And then there's a gene expression program that can start from which transcripts were stable. They're already ready to be translated so they can make those proteins, and those can cause downstream effects of either turning off a gene or turning it on by destabilizing or stabilizing the RNA. And you get this really complex interplay of a gene expression program.
I happened to do this recording like what, 3 days before you're retiring, which I didn't realize when we scheduled this call today. But what do you what do you hope that this paper will serve in terms of informing the larger hibernation community? Like, what do you think the larger hibernation community can take from this paper, not just for 13-line ground squirrel researchers, but for people studying other species as well?
Well, I guess I think that all hibernators are going to use the same basic genetic programs to achieve this, partly because I think that hibernators retain these traits from their ancestral hibernating way back when mammal. Right. The thing everybody needs to take away from this is that if you're going to do gene expression profiling, you're going to look for genes, you better use very carefully collected samples. It's not okay to, say, oh, this animal's been torpid, who knows for how long. Right. Because the gene expression dynamics are so, I think they're probably very stereotypic. And I think the other surprise, previous studies have generally used assemblies of transcripts to define what are the genes. Yep. And then they use the mapping of those back to known, like human, to annotate the genes. And there's been a complete bias towards looking only at protein coding genes because of that approach. And here we look at any transcripts that are defined in our data and map them back to genome. And in many cases, we find we don't know what they are. I mean, we managed to annotate a bunch that had not been annotated previously by the annotation pipelines that rely on homology. Yep. But still, there are a huge number of expressed transcripts. But there's a lot out there that we don't know what it is. And I think we illustrate that with one example. It's a transcript that is highly differentially expressed. It has no known homologue that we've that are pipeline annotated. But when you look at it specifically, you can see that it maps to the human genome. And it looks like a fractured gene that's been hit by transposons. Yeah, so, that might be what you'd expect for an important hibernation gene that has died over evolutionary time because it's not being used in a non-hibernator, but it's still there. And so there's a whole lot of comparative genomics that could be done, even taking these differentially expressed transcripts as a starting point to look for key genes of interest that could be then studied functionally.
Yeah, it'd be interesting too, as more genomes come out of the Broad work too. I know there's kind of another onslaught of genomes that Linda's working on with some of the groups at the Broad. That's like another 130 on top of the 240 or something that's been published. Be interesting to sort of look at these, quote unquote, broken or lost genes across those clades, right?
Yes, absolutely. One problem with this new genome, although the contiguity is great and it's really approaches the chromosome level of the 13-line ground squirrel, we still have all the ends that were there in the original sequencing. Yeah. So there are big tracts of ends in many genes, introns. I think our RNA-seq data covers that. And what I hope is that the genome can be re-annotated and maybe those even filled in because we have robust coverage. Right. Why not? Why does it have to come from DNA sequencing? Right. Yeah. So that would be another wonderful thing that could come from all this RNA-seq data.
Actually fill in those unknowns.
Yeah.
Exactly.
Yeah. Why should they be ends? They don't need to be.
Yeah, makes sense. What do you think is the next step? Like what do you think is kind of the next project that would come naturally off of this paper? Where do you think is the next step for this?
Well, I guess I still feel like the resolution isn't enough. I mean, I think there are genes that are worth looking at in our data set, the ones we were just talking about, maybe unknown genes. But honestly, better resolution would be even better. It would be nice to have more time points. I think we missed a lot in the gene expression program that unfolds rapidly during the rewarming, and a time point in there that was prior to our IBA time point would be really valuable. But in the brain, where the cell type complexity is so high, single nuclei should be used for the gene expression
that's what Katie is hot on too. She's going down that path with some of the samples we have. Is let's get down to the cell level and see what's going on, because it's so complex.
It is. It's too complex, and the really important gene regions or the really important functional regions of the brain that would be expected to be playing a role in this phenotype, right, by controlling body temperature and respiration and all those good things. We've averaged it all out.
Yeah.
So
thanks for getting on a call so quickly, Sandy, about this. It was great to just chat with you generally, because I haven't caught up in a while, and also get your perspective on the paper too. I really appreciate it.
All right. Well, thanks for your interest.